DNA is the instruction sheet for all of your cells and telling them how to replicate, constantly duplicating so you can do things like grow taller. Theoretically, that is. But how do the DNA replicate? That is the discussion for today, talking all about the replication of your DNA. Yes. In fact, DNA stands for deoxyribose nucleic acid, and this right here is just our sugar phosphate backbone. Our sugar phosphate backbone is actually made of, it's not really sugar, it's what we call deoxyribose, and then you have phosphate. Have 4 different nitrogenous bases right here. So we have adenine, which in our model that we're going to be seeing today is going to be orange. Thymine, which is in fact blue. We have guanine, which is purple, and it is a square. Cytosine, which is red. Those are our 4 bases. Also discuss something else very important. We'll go through this really quickly. Complementary is where strands are running all together in the same direction. That is complementary. Or you have anti-parallel, where it's simply going backwards. So, like that. So let's talk about replication and how everything is replicated. So step 1, I'm going to set up our DNA here for us. This is our DNA strand. Hydrogen bond right here. That's how everything sticks together. We then start our replication process. Introducing Helicase. This Helicase comes in and splits our hydrogen bonds. It breaks, unzips, whatever term you want to use for splitting apart things. And as it does so, it's creating this replication fork. Right here is our replication fork. Right there. That's a replication fork. We have introducing this Lego character here. This is our DNA polymerase. DNA polymerase is what allows new nitrogenous bases to be created. We only have a certain amount of these blocks that we can use. So let's pretend that the helicase has broken apart all of the bases. A polymerase will come in. It will create new nitrogenous bases respectively, allowing hydrogen bonds to be made. It will make another one. Whoops. Apologies. And there you go. Cytosine mends with guanine. And DNA polymerase proofreads everything. And then the inside is what ends up being your new DNA strand. And then what ends up happening is everything will then come back together because they still are matching and they are semi-conservative because it keeps what we already have and it adds in the new. So, thank you very much, guys, for joining me. And this has been how deoxyribose nucleic acid replicates.